Hi guys, this is Dr. Lorenz. This video is going to cover material from the end of Lecture 13 as well as the whole of Lecture 14. Just as a reminder, you are still responsible for all of the material that's covered in these lectures, so do make sure that you watch through the whole thing. So just as a bit of a review, we covered this in class, but just to help you remind yourself, get back into the swing of things. We started to talk about the differences between sensation and perception. So sensation is the registration of physical stimuli from the environment by the sense organs. So this is the capture of some sort of energy from the outside world by the receptors of our sense organs. And this is going to be very simple information, very gross level stimuli. Whereas so the dress here would be representing the very basic level sensory experience that we might see from a visual stimulus. You might see dark and light, or you might see some basic figure information, but it's still very gross level of information. However, perception is our subjective interpretation of these sensations combined with our expectations, our emotions, our experiences, learning, memory, attention, those sorts of things. So our percept here of this picture includes our subjective interpretation of the sensory information, as well as our expectations about what kinds of light conditions this dress is under very deep shadow or not deep shadow, and so on and so forth. So the difference between sensation and perception is that sensation is about the registration of some sort of energy from the outside world on your sense organs. It's a very simple level, whereas perception is our subjective experience and can include more than just sensation. So we saw this question in class. The answer here would be ingredients is to cake, right? Because you use the ingredients to make the cake, but the cake is made up of more than just the sum of its parts. Okay. So throughout the next few weeks, as we go through the various different senses, vision, hearing, smell, taste, and so on, we're going to see the same basic pattern play out across all of the different sensory systems. So some sort of sensory information, some sort of physical stimuli has to come in from the outside world. So this could be light hitting your eye. This could be a sound wave hitting your ear. This is some sort of physical energy is coming in from the outside world and is going to hit the receptors in your sense organs. It's going to be intercepted by those receptors. So your receptors, your sensory receptors, are specialized cells that are going to convert energy from the outside world into an electrical impulse, into an action potential. So these receptors are going to convert physical energy from the outside world into electrical energy in your nervous system. And this process is called transduction. So a receptor can be said to transduce energy, meaning that it's converting it from one form to another. So once information has come in and been received by the receptors, right? Once some sort of sensory information has entered into the receptors, then those receptors are going to send their information up to the thalamus, and the thalamus is going to determine what kind of sensory information that is. Is it visual information? Is it auditory information? And so on. And the thalamus is going to have special nuclei whose job it is to relay that information to the appropriate sensory cortex. So it might look at a piece of information coming in from the sense receptors in your eye and say, okay, this is visual information. So I'm going to send it to the visual sensory cortex. The first place that it's going to hit once it actually gets into the brain, where that information is going to go after it gets into the brain, is the primary sensory cortex. So the process of going from being information, physical stimuli in the outside world, to being transduced into some sort of action potential, to then being relayed through the thalamus. This whole process up until this point is still at the level of sensation, right? The information that is being converted is still relatively simple in its form. But once the information hits the cortex, then it's going to start being perception. So the steps prior to hitting the sensory cortex, to getting to the sensory cortices, is the encoding and the neural relay steps. And these are still within the realm of sensation. But once we actually get to the processing of that information, the combination of simple information to create more complex information, that's when we start getting into perception. So the sensory cortices and the association cortices are what are responsible for perception. So the primary sensory cortex within each sense modality is going to do the most basic level of processing. This might be combining two pieces of information together to create a slightly more complex piece of information. The secondary sensory cortex is still going to stay largely within the same sense modality, but is going to create somewhat more complex information, more complex processing. So for example, you might have visual information coming into the primary sensory cortex. The primary sensory cortex might be able to tell you what is light and what is dark, or whether something is a figure or that is discrete, or if it is blending into the background. So very basic level information. The secondary sensory cortex is going to do more advanced processing. So it might start to evaluate the contours of the shadows on a particular visual stimulus, or it might start to be able to process important information about the shininess or the colors and how they relate to each other within a visual stimuli. And then finally, the association cortices are where the different senses are going to come together. So when we experience the world, we don't experience our perception of sound differently and separately from our perception of vision, right? We experience them all together all the time. And so it's the association cortices that kind of merge everything together to create our final experience of the world, this association between different senses to create our overall whole experience of the world. So going back just a little step, our sensory receptors are specialized cells that will turn physical energy from the outside world into action potentials, into electrical energy. So each one of our senses have special nerve cells whose job is to turn some sort of physical energy into neural activity. So our sight receptors, our vision receptors are going to convert light energy, right, electromagnetic energy, into electrical impulses in the brain. Touch, somatosensation, is going to use mechanical energy, displacement of the flesh in some direction, or a sense of movement. So both of these would be related to movement or to mechanical energy. Hearing is going to rely on receptors that are going to be able to detect the movement of air molecules in space, aka sound waves. So this too is a form of mechanical energy. So both touch and hearing use mechanical energy, or they convert mechanical energy from the outside world into electrical energy in our brain. And then finally, taste and smell are going to use chemical energy. They're going to take in small amounts of chemicals associated with odorants, smells, or tastes, and is going to cause chemical reactions to happen that are going to drive the generation of action potentials. So in each of these cases, be it electromagnetic energy or mechanical energy or chemical energy, the sensory receptors are responsible for turning that physical energy that's coming in from the outside world into the electrical energy associated with an action potential. Also important to understand as we broadly think about all of sensation and perception is the concept of a receptive field. So a receptive field corresponds to a specific part of the world to which a sensory receptive organ responds. So it's whatever part of the world to which that specific receptor is tuned. So much in the same way that a player on the baseball field is responsible for the ball only when it is in that little section of the field, right? So in this diagram here, if the ball enters into the second baseman's area of the field, then they will interact with the ball. But if the ball is all the way over in left field, then the second baseman is not going to get involved, right? Because it's outside of their receptive field. So a receptive field is useful. Having a receptive field is very useful for sensory information because it helps to make it more manageable, right? It makes it such that an individual sense organ is only going to be, or an individual sense receptor rather, is only going to be responsible for a small sliver of information about a small piece of the world. It makes it much more manageable. And it also helps to locate where that sensory information is coming in from in space. So for example, in our visual field, if you imagine our visual field as just a flat space here, a flat kind of box, you will have sense receptors, visual receptors, that are going to correspond to each part of this field. So you're going to have one receptor that's going to be really important over here, and another receptor that's really important over here, and another receptor that's really important over here. And when you look at this visual field, these receptors are going to send different information to the brain. And so by determining which receptor is active at any one time, it tells us a little bit about where in the visual field that information is coming from. And so it can help us to orient sensory information in space. Another important concept is sensory acuity and sensitivity. So sensory acuity refers to your ability to detect differences within a sense modality. And your sensitivity refers to your ability to detect some physical energy registering at all. So for example, your visual acuity would refer to perhaps your ability to detect the difference between two very slightly different shades of the same color. Whereas your sensitivity would be your ability to see color at all. So both of these, both your sensory acuity and your sensitivity, have to do with the degree of density of the receptors within a receptive field. So how many receptors you have within a receptive field is going to determine the limits to your sensory acuity and sensitivity. So for example, you have many, many, many more tactile receptors on your fingers than you do on the skin of your back. So if we put two little pinpricks right next to each other on your finger, you're far more likely to be able to distinguish these two little pinpricks as separate stimuli than if we put them on your back, right? Because you have far, far, far greater density of receptors in your fingertip than you would have on your back. And the differences in receptor density are also going to determine the special abilities of certain animals. So for example, I've worked with a group of folks who research animal behavior. And one of the things they research are search and rescue dogs. So these are dogs that are specially trained to be able to detect people or specific objects. And to be able to detect even the tiniest little instance of any smell associated with that target. So for example, I've worked with a group who trains dogs to be able to detect human remains, or so human bones. And they're able to smell a sample of human remains even up to a mile underground. Right? So their sensitivity, their smell sensitivity is much greater than ours, because they have tons more sense receptors for smell than we do. If you make the analogy to vision, what you and I could see a third of a mile away, a dog could see more than 3,000 miles away, right? If we could compare the degree to which dogs are able to smell to the degree to which we're able to see. So they have about 10,000 times the odor receptors that we do, and this contributes to their ability to have much greater sensory acuity and sensitivity. So all of the sense receptors are going to connect to your cortex in some means, in some way or another. That does, if it's from your nose, from your feet, from your eyes, all of the sense receptors are eventually going to get to the cortex. So we're going to talk about this pattern of the receptors going to the thalamus, going into the cortex. And that is a simplification of what's actually happening. So there's no straight through point-to-point correspondence between one neural relay and the next. So each of these different levels is able to influence other levels of processing. Remember, the brain is going to process information both in parallel and in hierarchy. So, for example, at the level of the thalamus, these red boxes here, there might be multiple lines of sensory information that are coming in at the same time that are going to influence the perception, the processing of the other kind of information. So for example, here's a video of one of these kinds of effects. Let's see if I can make this play.
Have a look at this. What do you hear? Ba, 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 ba. But look what happens when we change the picture. Ba, 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 ba. And yet, the sound hasn't changed. In every clip, you are only ever hearing ba with a B.
So this is an example of how information that's coming in from one sensory modality, in this case from vision, is going to change the information processing of another sense modality, in this case, the auditory information. So if you see somebody's lips saying, creating the shape that would be associated with that person saying either ba or fa, it's going to change how you interpret those auditory stimuli. So you can even have new information that's going to come in that changes the interpretation of information that's already come in, that's kind of further up the chain of processing. So, for example, you might have some piece of information some piece of auditory information that starts to come in and starts to get, it goes through the thalamus, it starts to get processed through the primary sensory cortex, and now it's being sent to the secondary sensory cortex. So it's already actually gotten pretty far along in its processing. Now a piece of visual information comes in, a little bit delayed, but it changes how we might possibly interpret that auditory information. So like you might see somebody's mouth making a particular shape. And that will sort of jump the chain pass through all the way up into the secondary sensory cortex and change the interpretation of that original piece of information. So you can have new information that is coming in that jumps the chain, so the thalamus sends it directly into, say, the secondary sensory cortex, that is going to change how old information is being processed. So another example is if you see an object on the table, say, if that object starts to move, you will change your interpretation of that object from being just a stationary object to potentially a living object. And that's because this perception of movement is going to jump up the chain of visual processing and change your interpretation of what kind of object this is, even though the basic sensory information of this is a small object, this is a large object, this is a green object, this is a fuzzy object, whatever, even though all of that basic processing has already gone through. Okay. Now, one implication of the fact that our sensory relay is incredibly complex, our perception is incredibly complex, is that you can have illusions. So illusions are byproducts of our adaptation, our evolutionary adaptations to tend to interpret certain sensory stimuli in a particular way. So we experience whatever is the most adaptive thing for us to experience. We have evolved to have a subjective interpretation of sensory information, whatever subjective interpretation, whatever perception is most adaptive for our survival. So whenever we have a case in which the perception doesn't really match up 100% to what's being presented, it's usually because what would be true 99.99% of the time would be relevant to some aspect of our survival. So here's an example of that. So this is a visual illusion here. So just take a look at this figure and try and just sort of write down maybe on your notes or just think about what do you see in this figure. So just take a look and try and figure out what kinds of shapes do you see in this figure. And specifically, how many circles and how many triangles do you see in this figure? Just count how many circles and how many triangles. So you should see 3 circles, right? 1, 2, 3. And 2 triangles, right? 1 and 2. Do you see those? Okay. Turns out, if you just look at the level of what visual information is being presented, there are no circles and no triangles in this figure, right? So how do you see, quote unquote, see a triangle, despite the fact that there's no actual triangle here? Well, it's a byproduct of how our brain is creating a percept. So your brain is making some interpretations of the sensory information. It's going to kind of artificially create a sense of closure of things, of visual information to create the percept, the subjective experience of a closed shape, a closed figure. So a closed figure is being implied by these lines. And 99.9% of the time, when we see this kind of visual information, when we see a line that stops suddenly but picks up on the other side, it's because there is something in front of a closed figure, right? There's this triangle actually exists, but then this other triangle is on top of it and blocking our vision of the whole thing. So most of the time, that would be the case. That would be true. In this particular instance, it's not, because this is an artificially created figure. Here's another example of how illusions can occur as a byproduct of our evolutionary adaptation. So this circle here, take a look in the center here, and then take a look on the sides and kind of follow along around the outsides of this circle. And you probably will see this circle as spinning or moving, but this is actually a static image. There's no movement. This is not a video. This is just a static picture. And we perceive it as if it is moving, as a byproduct of how we perceive the edges of things. So in the real world, when the edges of visual information are a little bit blurry, that's generally because because that object is moving. And so we interpret objects in which the edges of things are blurry as, okay, there's movement happening there. 99.9% of the time, that would be the case. And it's very, very adaptive from an evolutionary perspective for us to be biased in terms of seeing movement, even when sometimes it's not there, because movement is incredibly important for understanding where there might be predators, where there might be prey, and so on. So in the real world, there aren't a heck of a lot of blurry edges like this. This is a very artificial image. So because in the real world, it would be adaptive to see this kind of visual stimuli as movement, that's how we have evolved. Here's another example of how illusions are byproducts of our adaptation. So take a look at this picture and see how many faces do you see in this picture? Take a look and try and count. How many different faces can you find? So the answer here is there are about 11 different faces that the artist depicted, depending on how you interpret it. So here's a face here with the eyes, nose, and the mouth. Here's a face here with the eyes, the nose, and the mouth. Here's a face here, eyes, nose, mouth. Another face here, another face here, another face here, another face here, and so on and so forth. And obviously, face on the cowboy. So if you look harder at these different quote-unquote faces, you'll see that none of them actually are really truly faces per se. They are collections of objects that will sort of induce the sense, or induce the percept rather, of a face. And this is because we are well adapted to pay a lot of attention to human faces. It's extremely important for our survival to be able to interpret and perceive very subtle changes in facial expressions. It's incredibly important to our social survival, to our survival as a species. And so we have evolved to spend a lot of time, a lot of attention, paying attention, a lot of attention to faces. We spend a lot of resources perceiving faces. And so anything that might remotely look like a face gets extra visual attention paid to it. And again, that's adaptive. So each one of these examples is intended to highlight the fact that we will experience the world in whatever way is the most adaptive for our survival as a species. How we perceive the world is dependent on what has been adaptive for our ancestors. How our brain turns sensation into perceptions is going to depend on what has been adaptive in the past. Our brain is going to perceive what has been the most adaptive regardless of what the actual sensory information coming in may have been, regardless of what's happening out in the world. Okay, So one of the reasons that we start talking about the visual system out of all of the different senses is because the human brain has a lot of space and time and energy that it devotes to processing vision. Vision is the number one sense for humans. Far more of the human brain is dedicated to vision than for any other sense, but that's not true for other animals. So, for example, dogs and rats have much larger of an olfactory bulb. So this picture on the right-hand side of your screen is a depiction of the olfactory bulb, that is the part of the brain that processes smell, in various different animals. So if humans have 1 unit of olfaction, then a mouse would have more than 2.5 times the amount of brain space that is devoted to that. So we devote an entire lobe to visual processing, right? The occipital lobe. And that's not including all of the parts of the brain that are dedicated to identifying objects that we see, or associating them with memories, or determining the emotional content of the visual information. For example, judging facial expressions and determining what emotion is associated with that, right? Those are all things that are related to visual processing that are outside of the visual lobe. So you can see there's quite a lot of the brain that is devoted to processing vision. In other animals, there's relatively more brain that's devoted to processing different senses. So thinking about that, what do you think about the answer to this question? True or false? A blind rat's brain will be more similar to a sighted rat than a blind human's brain would be to sighted humans. True or false? And I'll note that usually when I ask this question, at least one person will ask, well, is it any different if you are blind since you were born or if you acquired blindness, right? If something came along and removed your vision. The answer to this question will be largely the same. If whether or not this question is true or false will be the same if regardless of the kind of blindness, because whether you grow up without the ability to see and your brain develops without the ability to see, or you have an injury and your brain has to rewire and remap and be plastic around that injury, the result is going to be largely the same when we think about trying to compare rats and humans. So give it a moment to write down your answer. Think about the answer. So we have a little video here that will briefly orient you to the anatomy of the eye. This is just an overview. We're going to go through everything in a good deal more detail, but just so you have a broad perspective when we go in.
The location of the eyes enables us to see length, width, depth, and distance of an object. The eye has 3 layers: the sclera, choroid, and retina. The sclera is the outer white layer that maintains the shape of the eye. Muscles attached to the sclera control eye movements. Choroid is the middle layer that contains the blood vessels. The cornea is a clear circular area in the sclera where light enters the eye. The pupil is the circular opening in the front of the choroid. The iris is the colored smooth muscle surrounding the pupil, which adjusts the size of the opening according to the brightness of light. The lens is located behind the pupil and between the anterior and posterior chambers. The lens is a transparent, flexible biconvex structure that bends or refracts light rays, so they focus on the nerve cells of the retina. The chambers are filled with a watery fluid that gives shape to the eye and helps refract the light rays. The anterior fluid is called the aqueous humor, and the posterior fluid is called the vitreous humor. The retina is the inner layer and contains the nerve cells, the rods and cones, and the bipolar cells. Rods are sensitive to light but do not sense color. Cones sense color. The highest concentration of cones is in the fovea centralis. The rods and cones synapse with bipolar cells, which then synapse with the ganglia cells, whose axons form the optic nerve. Light rays enter the eye through the cornea, pupil, and lens, where the light rays are bent or refracted to focus them on the rods and cones, which transmit the stimulus to the optic nerve and then to the occipital lobe of the brain for interpretation.
All right, there were a lot of terms there, so let's go into each one of them in detail. So firstly, you have the cornea. This is the clear outer covering that surrounds the eye. This is the part where those of you who wear contacts, you actually press into that. That's where your contacts sit on top of. And those of you who wear contacts, you are superheroes, because I could never touch my own eyeball like that. Just behind the cornea, you have the iris. And so the iris is actually a muscle. In fact, it's the only muscle in your body that comes in multiple colors. If you've ever wondered where your eye color comes from, it actually comes from the melanin in your muscle tissue. So it's the only muscle tissue in your body that is pigmented. And melanin is just one color. It's kind of a brownish color. So it's the same thing that makes different skin colors. So you can imagine all of the different skin colors. They go from very light to very dark, and they're all kind of variations on shades of brown. So if that's the case, how can there be blue eyes or green eyes, or things that aren't brown eyes, given that it's a brown pigment that's causing the iris to appear colored? Well, it has to do with how that pigment is stratified within the cells of the iris. So if you have just a little bit of pigment in individual cells kind of patterned here and there, but not all in one block, then that's going to scatter light as the light goes through and bounces off the eye. And if you have that kind of scattering effect, the blue wavelength is going to be scattered more than the other wavelengths. This is called the Tyndall effect. It's the same reason why the sky is blue, even though there's no blue pigment in the sky. It's because blue light tends to get refracted in a different way than other colors of light. So the blue light, the blue color of eyes, or the green color of eyes, depending on the degree of pigmentation, has to do with how light bounces off the iris, which is why blue or green or gray eyes that are partially pigmented, appear to be such different colors under different lighting conditions. You may have heard someone say, oh, my eyes are sometimes this color, but in the middle of the day, they're a different color. Well, it's because different amounts of light are coming onto the iris. It's also why people with darker skin tend to have brown eyes, because the underlying gene for the degree of melanin production is the same for skin and for eyes. And this is also why albinos who lack the gene for melanin have red eyes. Can anybody think why that might be? Take a second to think about that. If we don't have any pigment in this muscle, what color would the muscle be? Well, it would be red, right? Because all you're seeing is the muscle, the flesh itself, and the blood vessels that vascularize that muscle. So that's why folks with albinism mutation, where you don't have the gene for melanin, have red eyes, as well as very, very light colored skin. Okay, enough about eye color. What is the purpose of the iris? Well, the iris muscle is going to open and close to allow more or less light in. So this helps to keep the receptors of the eye, the photoreceptors, active but not overactive. Right? Essentially, it helps to focus the information and not allow too much light in. So the hole in the iris is called the pupil. So the iris will work to open or close the pupil to allow more or less light in. You guys have seen this happen when you go from a light room to a dark room. Your pupil will change in diameter to allow more or less light in. And here's reason number one why ninjas are awesome. Right, so the iris is a muscle, right? Like all muscles, it can be trained. And so there are legends of how the ninjas used to train their irises to be stronger, to be quicker, to be able to adapt from going from a light to a dark environment. So they would flash light and dark like a strobe light. And over time, their ability to to refocus when they're moving from shadow into light or vice versa was improved. So they were able to be in a bright environment and then disappear into the shadows, and immediately their pupils will adjust much quicker than the people who were chasing them. So it just seemed like they disappeared into the shadows. All right. So next, going into the eye from the outside, we have the lens. So the lens is a transparent, flexible tissue that is going to change shape. Essentially, it's going to bend to help focus the retina on objects at different distances to us. And this is a process called accommodation. So accommodation is when the lens bends in different ways to help focus the retina, that is the space on the very back of the eye, on objects at different distances. So here's a way to remember accommodation. I'm going to do this exercise along with you guys. So take a moment, hold up your index finger about a foot away from your face, and you're just going and try and put it right where you have a wall behind you. So you're going to focus in on your finger here. Okay, so you're staring at your finger. It's about a foot away from your face. Now, as soon as your eyes adjust, you're going to look at the wall in front of your finger without moving your head. And as soon as your eyes adjust to that, you're going to switch back to your finger. So without moving your head, switch your focus from your finger, which is close to you, to the wall that's farther away. Okay? So if you go back and forth like this a couple of times, you'll notice it takes like a second for your eyes to adjust, right? It's a little bit blurry in between those moments. And you'll actually feel your lens. If you pay attention, you can actually feel inside your eye the lens shifting its shape. And that was your lens busy at work trying to make sure that you could focus at different distances. When folks are nearsighted or farsighted, meaning that it's easier for them to focus on things that are either farther away or near, that's usually because the lens has been either developmentally, they just grew up this way, or over time has become warped in such a way that it has difficulty bending in the right shape as to focus the light from a particular distance. So you'll notice with natural aging that folks become more and more nearsighted, meaning that it's easier for them to see things that are near, but much harder to see things that are farther away. And that's partially because as we age, this lens, this tissue, firms up and it becomes much harder for it to stretch itself out in the way that it's necessary to focus on things that are farther away. All right. So now we've talked about all of the pieces of the eye that help to focus light and shape it and bend it in such a way as to hit the retina. So what the heck is the retina? Why is it so important? So the retina is where light energy initiates neural activity by interacting with photoreceptors. Wow, big phrase there. Let's go over that again. Where light energy initiates neural activity. So this is the point at which that energy, remember we talked about how different sensory receptors are going to turn mechanical energy, photic energy, auditory energy, or chemical energy into action potentials. So the retina is where this happens when it comes to light. It takes light energy and it turns it into action potentials by interacting with photoreceptors. And the part of the retina where the photoreceptors are most dense is called the fovea. So the fovea is the middle part of the retina, in the very center of the retina, where you have the most photoreceptors, the most vision receptors. So remember, back to our discussion of the receptive field, that if you have the most photoreceptors in a particular place, this is where the visual field is going to be the most sensitive, right? It's going to have the highest acuity. There's also a part where all of these different photoreceptors collect together, they all synapse together and create essentially kind of like a superhighway, and that becomes the optic nerve that leaves the eye. But the spot where the optic nerve exits the eye to go into the brain, where the blood vessels enter into the eye as well, that part has no photoreceptors, right? Because it's just nerve projections that are leaving the optic nerve. There's no receptors there. It's all just axons that are leaving the eye. And so because there are no photoreceptors in that area, there is a blind spot, right? There is a literal blind spot. You cannot see anything in your visual field that is hitting that part of the eye because there's no receptors to see it. So here's an example of your blind spot. We're going to try and find your blind spot here. So you can try, if you have a laptop, you can try picking up your laptop and moving it around. If you don't, you're going to have to move your head around. So try to fixate your eye on the cross here. So you're going to cover up one eye. You're going to fixate on the cross. Just keep your eyes on the cross. And you're going to move either your computer or your head back and forth until you hit a point where that dot disappears. If you fixate on that cross, there will come a time where that dot totally disappears. And when you find that spot, go ahead and uncover that eye, and the dot should reappear. Now, keep your head exactly where it is. Focus in on the spot. Close your other eye, and the cross should disappear. So the reason why either the cross or the spot disappears is because it's in your blind spot. It's in the part of your eye where there are no photoreceptors. Now, why is it that when we walk around in the world, we don't see a great big hole in the middle of our vision? Well, it turns out what we perceive in the world, what we experience as seeing the world, our brain fills that in for us. It makes assumptions about what is probably in that spot based on the visual information that's coming in from the surrounding photoreceptors. And so it says, okay, probably in this spot, there's more blank white space because that's what everything around this cross or this spot are. It's just plain blank white space. So because I can safely assume that probably the thing that I'm not able to actually perceive is more blank white space, that's what it fills in for you. But your brain's doing this all the time. Every moment of every day, when you look around the room, your brain is making assumptions about what is in your blind spot and filling that in for you. All right. So, moving along. We see upside down. So when light is refracted through our lens and onto our eye, it is projected onto the fovea, onto those photoreceptors, upside down. And it does that simply because every time that you refract something through a lens, through a single lens, it will flip it upside down. The reason why magnifying glasses work is because they're two lenses, and so it flips and then flips back. Anyways, our lens flips things upside down, but we don't see the world upside down. Why is that? Well, again, at the level of perception, our brain flips everything that we see upside down again. But this is happening at the level of perception, not at sensation. What we sense is upside down. What we perceive is right side up. And there was actually a classic experiment in which the participants were given a set of glasses that had mirrors in them. So everything that they saw was flipped for them, was upside down. And so for the first day or two wearing these glasses, they saw everything in the world upside down. As you would imagine, they're looking at everything in a mirror. But then, after about 2 days, the brain of those participants realized, okay, the information that I'm getting is not correct. It's not mapping onto the real world. And so it flipped their perception. It stopped It stopped doing this flipping on the back end. And so everything they saw after those couple of days was right side up. Now, nothing was changing at the level of what their eye could sense. It was exactly the same information. But what was changing was that the visual system in the brain, the perception of that information was getting swapped over again. When they took off those glasses after a couple of weeks of wearing them, when they were just walking around the world, in the everyday world, again, took them a couple of days, but eventually their brain flipped it back. So this helps to demonstrate the fact that this flipping happens at the level of perception, not at the level of sensation. And you can actually test this for yourself a little bit. So try gently, very gently, pressing on the bottom side of your eyeball through your bottom eyelid. So just kind of press your finger up into your open eye on the bottom side of your eyelid. And you might be able to see a little black spot appearing on the top of your vision, right? And that's showing that when you interfere with the light coming in on the bottom, it's getting flipped in what you consciously perceive as coming in from the top. The retina and the fovea. So the retina is a light-sensitive surface at the back of the eye that consists of photoreceptors and neurons. This is a diagram of the kind of basic structure of the retina. Now you'll notice that the receptors, the actual parts that transduce, that change electromagnetic energy, light, into action potentials, are at the very, very back of the eye. And in front of them are several other kinds of cells. These are interneurons that will help to integrate information that are coming in from individual photoreceptors. But the light is coming in from this side here, and it actually has to travel through, filter through these other cells before it hits one of these receptors. So there's something that kind of interferes with our ability to get light information directly, except in one part of the eye, and that is in the fovea. So the fovea is at the very center of the visual field, and this is a receptive field that is specialized for very high acuity, and it's specialized in two ways. Remember, every time that there's a specialized function, like high acuity, there's got to be a specialized structure associated with it. So the fovea has two ways that it is structured to create this high acuity. So the first is that it is a part of the eye where there are very few cells that are sitting on top of the photoreceptive cells. So you notice that it's pretty clear path for the light to get into the photoreceptive cells in the fovea. At other parts, there's a bunch of cells in the way. Okay, so that's reason number one why the fovea has such high acuity, is because there's very little interference with light getting to those photoreceptor cells. There's very few cells sitting on top of it interfering. Reason number two is that the fovea has the most cones. The most cones out of the entire receptive field, the entire visual field. What in the world is a cone? Well, there are two kinds of photoreceptors, two kinds of cells that can detect light energy. There are rods and there are cones. So your cones are responsive to very bright light, and they are responsible for color vision, and they have very high acuity for fine detail. So you have the most cones directly in the middle of your fovea. They're able to see in extremely fine detail. Now your rods, you actually have many, many more rods than cones in terms of number, and they are very sensitive under dim light, low light conditions. They're also very important in the periphery of your visual field. So this is why the center of your visual field has the best color vision and acuity, and why your peripheral vision actually works better at night under low light conditions. So here's a picture of the density of the rods and the cones in relationship to the fovea. So if the fovea is here at the 0 mark, right, you see the vast majority of your cones are in the middle of the fovea. And the rods, you have actually very few rods in your fovea in the middle, but you have way more rods than you do have cones. Okay, reason number 2 why ninjas are amazing and so cool. So the ninjas figured out the fact that you have a lot of rods and that your rods tend to be in your peripheral vision and that your rods work out well when you are in conditions of low light. So they figured this out, and so rather than going with the more traditional attack method of trying to sneak in from the side of their target when they're trying to attack at night, right? Kind of sneaking in on the periphery of that person's vision. They knew actually that's the way that we're most likely to be detected, because there's lots of these rods and in somebody's peripheral vision, and they're very sensitive under low light. So if we're trying to attack someone under low light conditions, we probably shouldn't come in from the periphery. Instead, we should come from directly in front of them. So they would actually try to find the center of their target's visual field and try to blend in to the shadows and then just pop up seemingly out of nowhere. So it would give them this impression that they kind of came out of nowhere. Isn't that amazing, you guys? Okay. So moving right along. Going back to thinking about your cones, you have 3 types of cones, and these are what are called the 3-cone pigments. So these absorb light over a range of frequencies. So it doesn't necessarily mean if you have one of these pigments that it can only measure light at this exact frequency. It actually measures over a variety of frequencies, but it is tuned to be maximally active at different frequencies. So there's 3 major kinds. And then there's the rods. So the 3 major kinds of cones. You have the S cones, and these are the quote-unquote blue or short wavelength cones. And they are particularly helpful for giving us information about bright light in the middle of the noontime. They are particularly important, we're going to learn later on, for helping us to determine when it's daytime. Why would we tend to see blue light as a signal of daytime? Think about it for a second. Why would we see blue light as a signal of daytime? Because the sky is blue, at least on sunny days. So when we see blue light, our brain interprets this as meaning that it's daytime, and that helps us to regulate our sleep-wake cycles. People who lack this kind of cone actually end up having a lot of sleep difficulties because their brain doesn't have as easy a time figuring out what time of day it is. All right, moving along. You have 2 other kinds, the M cones, the medium, and then the long wavelengths. So the medium is green and the L long is kind of more reddish. And believe it or not, by combining all of these, you can get almost all of the colors in the visual spectrum. You have approximately equal numbers of red and green cones, but you actually have relatively fewer blue cones. But what's even more interesting is that even though we have fewer of these blue cones, of all of the kinds of cones, these are the ones that are the most spread out. So it's definitely true that for all of your cones, that the majority of them are going to be in your fovea. What few cones you have that are not in your fovea are going to be of this S cone, this short wavelength blue type. Okay. And then the rods are tuned to kind of somewhere in the very middle here, and they're really not focused on any particular wavelength of color, not focused on any particular color. They're really focused more on detecting light and dark patterns. Now, we only see a very tiny fraction of the electromagnetic energy spectrum that is around us at all times. That's what we call visual light. But as you can see, there's much more electromagnetic energy out there. So our photoreceptors, our rods and our cones, can only detect light that comes in from about 400 nanometers, which is perceived as a kind of a violet, dark purple color, to about 700 nanometers, which is perceived as a bright red. Now, that's not true for every animal. Other animals have structures that let them take in other wavelengths, such as receptors that can take in ultraviolet light. So, for example, this is what a dandelion looks like to a honeybee. Now, take a moment to think about why would it be adaptive for, or why would our cones have evolved to detect only the frequency band with this tiny frequency band of visual light, but not things like ultraviolet light the way that a honeybee can? What do you think? Why would we have not evolved the same visual system as the honeybee? Take a second to think about that. Well, basically, again, every time that we have an evolutionary question, we can usually say that we evolved that way because it was adaptive for us to do so. Our color system evolved to allow us to see more than other animals who are competing for the same resources. So, for example, being able to see the color of fruit versus leaves was very adaptive for us to be able to identify a food source that we could use, right? Like fruit. We can all eat fruit. But we're not able to really use the food source that a honeybee can use. We can't eat pollen. We can't use the nectar of a flower like this. So the honeybee has a wider range because it relies on the ability to see those sources of nectar. But we weren't able to get at those sources of food, so it wasn't adaptive for us to develop that visual system. Okay. So let's talk a little bit more about the retinal neurons. We've talked about the photoreceptors. Now let's talk about the different kinds of neurons. So there are 4 basic types of retinal neurons. There are the bipolar cells. The bipolar cells are going to receive information from the photoreceptors. So they're going to connect information from individual photoreceptors together. So these bipolar cells are going to take information from 2 or more of these photoreceptors, rods or cones, and they're going to combine them together. There are horizontal cells. The horizontal cells are going to link up photoreceptors and bipolar cells. So they're going to actually connect across a much wider range of cells. They are really functioning to coordinate the information at the level of the photoreceptor. But you'll notice the horizontal cell is not sending information up the chain. It's really focusing on processing information, basic sensory information coming in from these different rods and cones and bipolar cells. So one of the purposes of the horizontal cell is to help coordinate if cells on one side are very, very active and cells on the other side are very, very inactive, that it can help to either stimulate or inhibit the stimulation of other cells depending on the patterns of activation within the photoreceptors. Okay. Next up, you have the amacrine cells. So the amacrine cells, similarly, they're going to link together the bipolar cells who are connecting different photoreceptors and sending that information further up. And they're going to kind of coordinate the processing of information between the bipolar cells and the ganglion cells, the retinal ganglion cells. But you'll notice again, the amacrine cells are not necessarily listening directly to the photoreceptors. They're listening just to the bipolar cells, and they're coordinating between the bipolar cells and the retinal ganglion cells. So their purpose, again, is to help do some of the sensory processing at this level. And then finally you have the retinal ganglion cells, and these are going to be coordinating and kind of condensing the information that they're getting from the bipolar cells and to a lesser extent the amacrine cells. It's going to be combining all of that information together and then linking up together to form the optic nerve. So the group of these ganglion cells, the projections of these ganglion cells that all come together towards the back of the eye are what's called the optic nerve. Okay? You'll notice there's way more rods and cones than there are going to be retinal ganglion cells because the individual signals from these rods and cones are going to get condensed at the layer of the bipolar cells, and those are going to get further condensed at the layer of the ganglion cells. Okay. Moving right along. So now we've got all of this information, all of this raw sensory information that's being connected together in the axons of the ganglion cells into the optic nerve. And we're going to start to move into the brain now. Hooray! So we're going to move from the level of the receptors into the thalamus, into the thalamic relay nuclei. So there are three major pathways that visual information is going to take via the optic nerve into the brain. So the dominant pathway, the direction that most visual information is going to go, is through the geniculostriate pathway. And we're going to talk about that in a little bit more detail in a moment. But this sends information through the lateral geniculate nucleus into the striate cortex, which then is going to go to other visual areas. A much smaller pathway is the tectopulvinar system. So this is a system that actually is going to kind of bypass the primary visual cortex. And it's going to let information go directly from the retina into the parietal and the temporal cortices, which helps to coordinate the processing of auditory information or movement information. So as you hear something and you see something at the same time, it's important for you to be able to coordinate those two sources of information together. What you see is literally going to change how your brain processes the auditory information that comes in at the same time. And similarly, you need to be able to have visual information while you are processing a movement to be able to know that that movement is going as planned. And so this tectopulvinar system, which goes through the superior colliculus, is going to send information, kind of bypass the main processing, the main visual processing, and send it pretty directly to the temporal and the parietal cortices. And then finally, you have a third pathway, which is the teensiest, tiniest pathway, and this is called the retinohypothalamic pathway. And this sends light information directly from the eye, from the optic nerve, into the hypothalamus, into a special part of the hypothalamus called the suprachiasmatic nucleus. So you remember I mentioned that you have a couple of blue cones that sit just on the periphery of your vision, and these cells are going to project information into the suprachiasmatic nucleus, which is super important, as we're going to learn towards the end of the course. The suprachiasmatic nucleus is super duper important for helping to regulate our biological rhythms. So by using information from these blue cones in the periphery of our eye and sending it through the retinohypothalamic nuclei into the suprachiasmatic nucleus, it helps our brain to be able to regulate our biological rhythms, our sleep-wake cycle, and so on, using information about light. Okay. So as the optic nerve is transmitting into the brain, it is going to pass through what's called the optic chiasm. Optic chiasm. So the optic chiasm is the junction of the optic nerves from each side of each side of the face. So the axons from the nasal side, so the retinal ganglion cells, the axons from the nasal side, are going to cross over the optic chiasm to the opposite lateral geniculate nucleus. Right, so you can see here the nasal side is going to cross over to the opposite side's lateral geniculate nucleus. And the axons from the temporal, or the outer half, are going to stay on the same side and go to that side, the same side's lateral geniculate nucleus. Now, why would it be called the temporal side? Think for a second. What's this lobe here called? That's right. This is the temporal lobe, the one that's next to the ear. So the temporal side is the side that's on the outside, right, away from the nose. That side is going to stay, the axons that come from that side of the visual field are going to stay on the same side. So another way to think about this is that your nasal information is going to cross over the optic chiasm. The outside of the eye is going to stay on the same side. So information from your left visual field is going to go to your right brain. And information from the right visual field is going to go to the left side of the brain. So once it's actually in the brain, it's going to go into the thalamus, and specifically the lateral geniculate nucleus, before it's then sent through optic radiations into the primary visual cortex. Where is this? It's kind of at the very, very back of the brain. What lobe is this? That's right. That's the occipital lobe. So thinking about how information from the outside, how that transmits, information from the inside of the visual field, how that crosses over, how things from, the left side of your visual field is going to go to your right brain, and things from your right side of your visual field are going to go to your left brain. So thinking about all of that stuff. Think about a lesion at which point, so if we cut the nerves, at which point, which would create this visual field deficit? So this is a picture of a visual field deficit in which you cannot see this section that's blacked out, but you can see this section that is on the inside. Which of these, here, 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 or here, which of these would cause that kind of damage? Take a moment to think about what you just saw about crossing over, about the right and the left visual fields, and try to figure out what would cause this kind of deficit. Give it another couple of seconds here, and then I'll give you a big hint. There's your big hint. It's basically giving you the answer to the question here. If I were you, I would look closely at this diagram as you're studying, because it can be very useful in remembering precisely how these different parts of the eye transmit information into the different parts of the brain. Okay. So we're going to move along now. We've gone through the lateral geniculate nucleus in the thalamus, and it's kind of projected into the occipital lobe. So once it's projected into the occipital cortex, once that visual information has gone into the visual regions of the occipital cortex, it's first going to end up in what's called V1, which is called the striate cortex. So the striate cortex receives input directly from the lateral geniculate nucleus of the thalamus, and the striate cortex is going to process the most basic aspects of vision. So things like determining the edges of things, determining the shape, determining the basic location within the receptive field. The very beginning of color perception and form. And then once that information has been processed by V1, the striate cortex, the primary visual cortex, it's then going to be sent into these secondary visual cortices, which are called the extrastriate cortex. So basically anything that is outside the striate cortex. By the way, striate just refers to how that part of the brain looks. If we stain it, it looks like it's kind of striped, like the cells are striped, but that just refers to how they look on a stain. The other parts of the visual cortex don't look like this, so we call them extrastriate or outside of the striate cortex. Anyways, these constitute the extrastriate cortex. These other visual areas constitute the secondary visual cortex. And these are going to process more complex aspects of color and form, and this is where you're going to start to have processing of motion. From these secondary processing areas, you're then going to have that visual information is going to be sent to other parts of the brain. And there's sort of two major visual streams. So from this secondary extrastriate cortex, and also some information from the primary visual cortex as well. Remember, the brain operates both in parallel and in hierarchy. You're going to have information that's going to project up through the dorsal stream and through the ventral stream. So the dorsal stream, remember, dorsal is going to mean up on the top here. This is going to be information that is going to go into the parietal lobe. And the parietal lobe is going to help process this visual information and determine how we need to interact with that information. So how action should be guided towards an object. How should the body move in response to this information, essentially? The ventral stream, on contrast, is going to go in the ventral part, so it's going to go on the bottom part, closer to our bellies, into the temporal lobe. And this is going to be what's called the what pathway. So the what pathway is going to be what actually identifies what an object is, basing this off of experience, trying to fit this to templates from prior experiences, working with the memory systems to be able to map this onto other objects, trying to name an object, right? A lot of our identification of an object is trying to remember, either kind of consciously or subconsciously, remember the name of an object. So obviously, if you're remembering the name, you want to be someplace close to the language systems, which are in the temporal lobe. So this stream, the ventral stream, is the identification, the what pathway. The dorsal stream going up into the parietal is the how. The way that I remember this is that the parietal lobe is primarily responsible for somatosensation, as well as partially responsible for movement, whereas temporal lobe is responsible for language, for naming things. So I just remember, how do you move in relationship to this visual information, and how do you name what you're seeing? Okay. So once the visual system has made a basic percept, a basic decision about what the characteristics of that visual information is, it's going to send that information on to other cortices to identify it based on memory, based on deduction and logic sometimes. And this is, excuse me. So the temporal lobe, like I just said, this is partially responsible for doing that. And it actually has particular areas that are going to be sensitive to different kinds of stimuli. So different aspects, different parts, different layers of the temporal lobe are going to be more excited by different kinds of stimuli. So there's a part of your temporal lobe that is extremely excited by symbol shapes, but not at all excited by hands. There's another part of your temporal lobe that's extremely excited by hands, but not at all by directions. So each of these different kinds of complex stimuli gets a special part of the brain that is responsible for responding to those kinds of stimuli. So there can be times, there can be deficits when you have a problem in one of these areas, but not in others. So if you damage just this little tiny part of the brain that's responsible for processing visual information about hands, but the rest of the parts are fine, you'll be able to see and perceive everything of every other kind of information, but when it comes to hands, you're going to be kind of largely blind to that. It's not that you're blind to the shape or the form of the hands. The basic sensation of hands is still there, but precisely what those hands are doing or what gesture they're making, for example, you would not be able to recognize. A kind of classic example of this, this is called an apperceptive agnosia. Excuse me. A kind of classic example of this is prosopagnosia. You guys may have heard of this. So prosopagnosia is the inability to recognize faces. So folks with prosopagnosia can see the individual features of a face. So they could tell you if the face of that person is light or dark. They could tell you if that face was big or small, but they would not be able to tell you that it is a face. So they would be able to look at a picture of a face and say, okay, this is a very dark stimulus, this is a very big stimulus, a very small stimulus, but I don't know what it is. They would not be able to tell you that it is a face. So I'll give you guys a little story about this. For a while, I worked in a hospital setting, and I was working as a psychologist in a trauma ward with a bunch of neurologists, including some neurologists who were in training. Every day that I worked there, all of the surgical residents, the nurses, the physical therapists, and so on, all of the MDs, they would all wear scrubs or lab coats as they did their work. And the psychologists, because we kind of operate in a slightly different setting, we would just dress in our normal kind of business casual clothes. And so I was working for a little while with a neurology resident, and every time that he saw me in the hallways, he would get really excited and he would say, "Oh, Dr. Lorenz, it's so great to see you. It's so lovely." And he was a pretty friendly guy, so this was not totally out of character, but he was really over the top about it. And about 3 months after working there and having this reaction every time that he saw me, "Oh, Dr. Lorenz, isn't it nice to see you today? Isn't the weather lovely?" No, it's not. We're living in Seattle. We were sitting in the break room, and we just got to chatting about how we got started in our careers. And I asked him what got him started in neurology. And he said, well, I got interested in this because I have this special condition called prosopagnosia. And as soon as he said that, it finally clicked for me why he was so excited every time he saw me. It's because he had such trouble recognizing people's faces that he had to rely on every other cue he had to be able to recognize who he was talking to at any particular time. And he was working in a hospital environment where everyone is in the same uniform, except for me, the psychologist, wearing different clothes every day. So he knew if he saw this person wearing business casual clothes in a hospital, that he knew exactly who it was. So he got so excited, he finally recognized someone. So that's prosopagnosia, right? The inability to recognize faces. Now, he could tell me. He knows that I have, kind of light-colored skin. He could tell me that I had kind of reddish-brownish hair. He could tell me I had short hair. He even could tell me the color of my eyes. But he could not tell me what my face was or who I was based on my face, because this part of his temporal lobe that was responsible for processing faces was damaged, or rather, in his case, it never developed. Another kind of example is that when I was working in the hospital and I was trying to help diagnose head trauma, so if there was head trauma to the side of the head because of a temporal lobe damage, this often happened when there was gunshot wounds or knife fights, things like that. When people get stabbed in the ear. It's really, really nasty stuff. I would ask people to name objects in the room, and I would ask different kinds of objects. So I would name a face, a hand, a pen, a simple line. I would draw directions, or I'd make things move around. And depending on what precisely they were and were not able to perceive, I could make some inferences about precisely where in the temporal lobe that damage may have spread to. Okay, so take a moment here, thinking about the visual processing parts of the temporal lobe and what the temporal lobe is primarily responsible for processing. Which of these images would cause the visual processing parts of the temporal lobe to activate the most? Give it another second. And I'll give you guys a little hint. Which of these images would require you to activate the most different parts of the brain, most different parts of the temporal lobe? What do you think? All right, going to move along. So, we're going to talk a little bit about how does the brain create different aspects of vision, and then we're going to end with how does the brain create color vision. So how does the brain create a spatial map? Well, the very simple answer is that it uses a map that's created by having many different cells spread out over your eye. So if you think of your eye, the retina, the visual field, as kind of being spread out in a box with your fovea here in the center, depending on where the light is coming in and hitting the different parts of the fovea, that is going to map onto different parts of the occipital cortex, where that axon projects to. So it's going to preserve that spatial map that is created by having a large receptive field within the eye. It's going to preserve the receptive field of the eye and project onto different parts of the visual processing centers of the brain. So the receptive fields of different retinal ganglion cells that are at different parts that are kind of lined up next to each other within the fovea, these different, depending on where they are, it's going to project into the receptive field of a single laterate geniculate nucleus. Remember, that's the part of the thalamus, which in turn is going to project to different aspects of the striate cortex. So the brain is combining this information from these lower order to higher order, more complex processing areas, but it's maintaining that original order all the way up. So the spatial ordering starts in your retina, that gets folded into the higher levels, all the way up into the brain. And this is how you locate things in your visual field, how you know that something that you see far off in the periphery of your vision is over to the side, and something that is in the middle of your vision is kind of right in front of you. How do photoreceptors see? How do we see shape? How do we detect outlines? So you have some retinal cell ganglion. Remember, these are the cells that combine information across multiple different photoreceptors. So some of these are going to serve as on-center cells. So an on-center cell is going to be maximally excited when there is light that is shining in the very center of its receptive field. But if there's any light that is shining on the outside, the kind of the periphery of its receptive field, it's going to be inhibited. And so it's going to do this by combining two or more photoreceptors that are next to each other. So one of which is going to fire when it's in the light, and the one that's next to it is going to fire when it is dark. So if you have both of them firing at if you have light that's shining on both of them, one of them is going to be excited, the other one is going to inhibit that. Right? And so when the two of those together, they operate together. So the only way to get both of them to be super excited at the same time is by having only the photoreceptor that is excited in the middle be excited, and then the other one, excuse me, be in the dark. When that happens, they're both firing. And that will be when that entire retinal ganglion cell is maximally excited. So if there is light shining only in the center of the on-center field, it will fire a lot. But if there's light shining on the outside, it'll be inhibited. So when there is an edge, a light-dark edge, right, it'll block some of the light coming into the periphery of these on-center cells. And so that is going to fire that retinal ganglion cell the most, right? And that information will be compared to other retinal ganglion cells in the same area to determine where that light dark edge is. So if the whole retinal ganglion cell here is in the dark, it won't be firing at all. The next cell over will be firing just a teensy bit, just a little teensy bit. It'll be inhibited from firing. The next one, who has some of it, is going to be firing just a teensy bit more, but the one that has its center being excited, and then as much of its periphery as possible being inhibited, that will be the retinal ganglion cell that's firing the most. And so the brain is going to look at the pattern of activity across all of these cells, A through E, and say, okay, based on the level of firing across all of these cells, this is where the light dark edge is. So remember, this goes back to our basic principles. The nervous system works by balancing excitation and inhibition. How does the visual cortex create the idea of shape or orientation, meaning how an object is lined up in space? Well, you have some simple cells that are going to detect orientation by being oriented in the brain in such a way that the retinal ganglion cells that project onto that simple cell will correspond to the orientation in the brain, right? So if there is if there is information that is projecting to these cells indicating that the light is not in that orientation, it will have no response. But if the projections to those simple cells in the visual cortex corresponds to its maximal orientation, it will have a very, very strong response. And then that too is carried forward into the different layers of the visual cortex, of the striate cortex. Okay, and how does the visual cortex create perception of motion? So in addition to your simple cells, you have complex cells. So complex cells are going to detect movement as well as orientation within a plane. But importantly, the complex cells don't have a way of detecting where that stimulus ends. So the complex cells are going to combine information across multiple simple cells with dendrites that are placed at a short distance from each other such that they play off the spatial and the temporal summation of those simple cells to make the cell most likely to fire when those simple cells, each of which is corresponding to a particular orientation, are fired in succession. So if this stimulus is moving down this way, you can imagine the simple cells that are corresponding to this part of the visual field, or this part of the receptive field, this part of the receptive field, this part of the receptive field, that there's different simple cells that are placed next to each other in this line. So if this one fires, and then this one, and then this one, and then this one, and then this one, in rapid succession, then this complex cell will fire. Right, so it's able to detect the movement based on the individual simple cells firing in rapid succession. Now, a hypercomplex cell, like a complex cell, is going to detect motion, but it is also going to have an inhibitory zone. So that helps to create the perception of the end of a moving object. So if the object extends beyond its receptive field, the hypercomplex cells firing will be inhibited. So when a hypercomplex cell is maximally active, it indicates that it is on the edge of a moving object. So it's going to work by combining input from both the simple cells and the complex cells in such a way that the activity of the simple cells are located at the edge of a receptive field are inhibitory. Okay? So these two things together help to create our perception of motion And essentially, they work by combining the physical structure of each individual neuron and their organization, which is what allows them to create their different functions. All right. Last little piece here is thinking about color. So, seeing color starts with the cones. So cones are our photoreceptors that are responsible for perceiving color. And what color we can see depends on what cones we have. So we have a different set of cones from dogs, for example. And so they see, and cats, they see a different set of colors, right? So this is what the rainbow that humans see. Dogs and cats also have cones, so despite what you may have heard, they can see color, but it's much more limited because they lack this middle cone that is responsible for detecting the difference between blue and green. So they see a much more muted set of colors. And similarly, if you lack a particular cone set, you'll have problems with different kind of color vision. So you may have seen these circles before when you were in school, and they may have been testing you. And these are designed to help identify if a child is lacking a particular set of cones. So if you lack the L cones, the red cones, the long wavelength cones, you will not be able to distinguish between these dots here and these dots here. They'll all look basically the same to you. Excuse me, that's part of what contributes to certain kinds of red-green color blindness. And here's a little interesting fact for you guys. There's some speculation that towards the end of his life, Van Gogh actually lost a particular form of his of cones, of the green cones. And this is a picture, this is a very famous painting, Starry Night. This is the actual version that Van Gogh painted. But this is the same picture if you filtered it in such a way as to give the color perception associated with green cones. So you can kind of see that the colors are kind of more of a traditional frame. This is sort of more greenish tint. And so there's some idea that he may have had a particular genetic disorder that caused the green cones to slowly die off towards the end of his life. I just think it's kind of interesting idea. So there's two basic theories about color vision. So the first is the trichromatic theory. So the trichromatic theory says that color vision is based on the coding of three primary colors: red, green, and blue, right? So red, green, and blue. Trichromatic, three colors. So the colors that we see are determined by the relative responses of the different cone types. So like in this image in which the lights are all mixed, if all three of these cones are active at the same time, at exactly the same proportion, we see white. If the blue cones and the green cones are active at the same time, we see this kind of light bluish green. If the green cones and the red cones are active at the same time, we see yellow, and so on and so forth. So some of the evidence for this theory comes from the fact that we do have cones that are sensitive to three different wavelengths of light, right? We have short cones, which are responsible for blue, medium cones responsible for green, long cones responsible for red. And color blindness, most forms of color blindness do seem to correspond to these. But there is a big part of the trichromatic theory, a big problem with the trichromatic theory, and that has to do with higher order color processing, such as the creation of after images. So I want you guys to take a moment, and I want you to look at this green and yellow flag, and you see that little dot there. I want you to kind of stare, not kind of, go ahead, stare at that dot. Fixate your eyes on the dot. Keep looking at the dot. Try your best not to blink too much. If you have to, that's okay, but really you're going to stare, you're going to keep staring, keep staring. Keep staring at that dot. Don't move your eyes away. Keep your head perfectly still. Keep staring, keep staring. Ta-da! Now if you keep looking in the same spot, you should see the American flag, red, white, and blue, Old Glory, in your after image. So opponent process theory describes why we see an after image. Ta-da! So the opponent process theory is it's an elaboration on trichromatic theory. It's an explanation of color vision that emphasizes the importance of the opposition of different colors. So opponent process theory says that we have red vision that opposes our green vision and blue vision that opposes our yellow vision. That there are certain retinal ganglion cells that will be stimulated by one of the pair, and inhibited by the other. So you have certain retinal ganglion cells that will be stimulated by red and opposed by blue, right? And inhibited by green, rather. So red, green, blue, yellow. So when we take a lot of time really deeply processing one of these colors, if we suddenly remove that stimulus, then the cell will very briefly fire in the opposite direction and create a color, a perception of color of the opposite side. So another piece of information that the opponent process theory helps us to explain is that our color does not actually respond to particular wavelengths necessarily, but rather we tend to create color at the level of perception. So an example of this is color constancy. So our perceived color is always constant relative to other colors, regardless of changes in illumination. Right? So if you put a lens over a field in which you have like a red lens that is kind of shading everything in a red shade, we still perceive everything within that red shade as having constant color relative to other colors. We would still see a red apple as having a different color than a blue cup, for instance. And if we took away that red shade, then we would still see the red apple different from the blue cup, right? So relative to two objects within the same frame, we see color as a constant phenomenon, regardless of the illumination. And what that suggests is that color is created, to some extent, at the level of perception, that our brain takes into account the context of the whole visual field to be able to determine the color of any individual object within it, rather than relying purely on the sensation. Because if we put a red screen, if I put a red color over the screen, right, the raw sensation that is coming in, the thing that is actually driving the photoreceptors to fire, would be all red. We wouldn't be able to see the difference in colors between things. So opponent process theory, it talks a little bit more about color vision as being reflective of perception. Trichromatic theory talks about color vision as being entirely due to elements related to sensation, our photoreceptors themselves. So trichromatic theory is a theory of color vision that talks about color as a function of sensation. And opponent process theory says that color vision is a function of perception and higher order processing within the brain.